Hello, everyone. I'm Chris McCurdy. We're going to just wait a couple minutes as people are sort of piling in. We'll just give them a couple of minutes to come on in, and then we'll go ahead and get started. Feel like I should dance or something so that there's entertainment. All right, the numbers keep going up, so we'll hang on here for another minute or so and let everyone join that is interested. All right. Well, I think in the best interest of time, we'll go ahead and get started. People may come in as we continue, but we'll at least get started with the introductory remarks and then um, turn it over to the panel discussion. Really like to welcome you to the 2024 Symposium on Substance Use Research. My name is Chris McCurdy. I'm a professor at the University of Florida, professor of medicinal chemistry. I also serve as our associate dean for faculty development. I've been doing natural products-based research, plant-based research since a PhD student, actually, in graduate school. So more than a couple of decades, almost 3 decades now, as a matter of fact, working on various plants throughout my some that might be known to some of you as Salvia divinorum, which was the source for salvinorin A, a hallucinogen. And then most recently, we've been working on Mitragana speciosa, which is the source of kratom or kratom, depending on like tomatoes or tomatoes. But that's essentially what my background is coming into this panel. And I do want to just make a couple of comments before we begin, and then I'll talk a little bit about the overall view of this session. So just a couple of reminders, we are recording this session, and we'll send an email to all the registrants and post on social media when the recording is available. Also, this session is a webinar, so all the attendees, please use the Q&A function to submit questions, and we will try to answer them as time allows. Um, First of all, I'm really excited and honored to be able to moderate this panel and have these three outstanding speakers with us today. These experts joining us come across a range of disciplines. So we have Dr. Jenny Wilkerson, Dr. Sandeep Nayak, and Dr. Shanna Babylonis, and I'll let them all introduce themselves in a moment. Before we do that, I just want to make a comment about doing work with plant-based drug-based medicines, and that's that I think we all have an understanding that the ones that are being targeted, particularly in substance use areas, are obviously psychoactive. But we many times have the misconception that because it's a natural product, it's safe, and it can be readily consumed without any regard to toxicities or side effects. And just want to point out that all of these plants that we'll talk about today have their unique life and not only are they being utilized to reduce harm, they can still cause harm, as can any substance. And perfect examples of these are just natural substances that we use and prescribe to this day, like morphine. Morphine we know can be very toxic and deadly if consumed in a high enough dose. The same needs to be considered when studying any plant material, and just a caution, a word of caution. And so most of the work that will be discussed here today has been looking at many of these substances in more therapeutic harm reduction aspects, and not in most of the toxicological or harm causing aspects. So some of that can come up, but just wanted to kind of put out that sort of preamble of what we're going to do. And with that, I'm not going to talk anymore. This is really meant to showcase our three experts here today. So, Dr. Wilkerson, could you describe your background, research area, and expertise relevant for this panel?
Thank you so much, Dr. McCurdy. Absolutely. So my name is Dr. Jenny Wilkerson. I am an assistant professor in the Pharmaceutical Sciences Department at Texas Tech University Health Sciences Center in the School of Pharmacy. And so I have been working with cannabinoids and natural products for about 16 years, ever since I was a graduate student. So I started off looking at the effects of cannabinoids in animals with the potential for neuropathic pain. And then I segued into looking at the role of the endogenous cannabinoid system for pain control. And so that was very fruitful, very a great opportunity that I had as part of my training. And then I actually moved to University of Florida, where I had met Dr. McCurdy, and I started working with his group and several other groups at University of Florida, looking at the natural product kratom. And so I've been looking at the effects of kratom for potential pain and substance use disorder therapeutics.
Thank you very much, Dr. Wilkerson. Dr. Nayak, can you share the same with us?
Sure. My name is Sandeep Nayak, and I am a psychiatrist. My clinical work is in addiction in Baltimore, so primarily opioid addiction. However, I spend most of my time doing psychedelic research. So am an assistant professor at the Johns Hopkins Center for Psychedelic and Consciousness Research, and the medical director also. And so my work there is primarily looking at the use of psilocybin, which is a psychedelic drug found in mushrooms, as a treatment. And we use a particular, therapeutic modality with it, but we're looking at it for broad array of psychiatric conditions, depression, substance use disorders, OCD, etc.
Great, thank you. Thank you so much. Dr. Babylonis, can you share with us as well?
Absolutely. Hi, I'm Shayna Babylonis, and I am at the University of Kentucky in the College of Medicine. And I'm an associate professor and also the director of our Cannabis Center. My research primarily focuses on the abuse potential and the therapeutic effects of cannabinoids, most frequently CBD and cannabis. And then I'm also very interested in the intersection of opioids and cannabinoids, like some others here. And all my work is in human participants, so it's all in the clinical trials or the human laboratory.
Great, thank you. So we have a real diverse sort of span from basic science all the way through clinical science and controlled clinical trials here on the panel. And so one of the things that we thought we would do is first really look into how people got interested in this area, and if they could address for us sort of some of the limitations or issues that they've run into in the field so far. Jenny, why don't we start with basic science?
Sure, absolutely. Thank you so much for that. So initially, I got started, as I mentioned, looking at the involvement of a specific cannabinoid receptor, the CB2 cannabinoid type 2 receptor, which is predominantly found on immune cells, and its involvement in inflammation and chronic pain. And so that was my dissertation work. And then I moved on to my postdoctoral fellowship, where I started looking at the involvement of the endogenous cannabinoid system and other cannabinoids that act on cannabinoid receptors and their involvement in pain. And unfortunately, about the time that I started doing that work, the opioid epidemic really started to take off. And so there has been a known amount of literature that has kind of characterized that, as Shanna was mentioning, the intersection of cannabinoids and opioids. And so I then started looking at, during those years, because the opioid epidemic really was starting to take off, the intersection of cannabinoids and opioid sparing effects for pain control. And maybe we can use cannabinoids to help in substance use disorders, including opioid use disorder. From there, as I mentioned, I moved to University of Florida and started working with kratom and Metrorhina speciosa with Chris's group. And so one of the major limitations kind of that was run throughout my research is not only the fact that I'm a basic scientist, so sometimes our models are not 100% translatable to humans. So we always have to take caution when we look at the results that we have in our experiments, because they may not be directly translatable to humans. And so we always want to be cautious. When we interpret our data, we don't want to claim that a drug can, or that our compounds can cure things or treat things in humans, because we simply don't know that based upon what we find in animals. So that's one major limitation of the work that I do. Another major limitation that Dr. McCurdy and I have spoken at length about is the fact that most of our research is in a very reductionist form. So a lot of our research is looking at specific compounds found in natural products. And so when humans are ingesting and taking these products, they're not specifically taking that one compound. They're taking the whole plant or the whole substance. And unfortunately, with the way a lot of our research goes, we mostly study things on a very reductionist compound-based approach, because we have to figure out what is going on with each individual compound before we can put them together to actually really decide what each compound is doing in concert with one another.
Yeah, I think that's really a key piece. And I think the other limitation from just a basic science, natural products perspective is availability. So, we when we're studying a new plant or a new extract, if you will, getting enough material to actually study becomes very problematic. And Jenny, maybe you could address real quickly what we know about and what we don't know about the kratom alkaloids. Most of the focus has only been on really two compounds.
Yeah, thank you so much for that. So with kratom, because it is a field of research that's really emerging, so we know that the kratom plant, the two major alkaloids are metrorhinin and 7-hydroxymetrorhinin, and we know that those two alkaloids produce a lot of the kind of psychomimetic effects of the drug. But just like with cannabis, we know that metrorhinin contains numerous different alkaloids, terpenes, and so we don't really know what all of those individual compounds do, and it's hard to approach those other compounds in a really mechanistic deep dive, because as Dr. McCurdy alluded to, kind of isolating and synthesizing those compounds in a meaningful amount that we can actually test in animals becomes very problematic. And so we're really kind of limited to by the tools that we have to really examine the overall impact of those other compounds that are comprised of the whole plant.
Yeah, thank you. Thank you for that. And I think it's a nice segue into Sandeep, who's doing things with really an isolated natural product. And maybe Sandeep, you can share how you got interested in this area and really some of the challenges and limitations that you've faced with working with a Schedule One substance in particular.
Sure. I had a sort of random set of interests that kind of ended up congealing. So I was a religious studies major in college, interested in studying Buddhist mysticism. I was involved in harm reduction work, like with injection drug use, and also did some meditation, like lab research, and discovered, decided to go to med school, discovered psychiatry, addiction psychiatry, and then these interests began to combine. The psilocybin work that Hopkins does, I mean, this is now part of a really big, there's billion-dollar drug companies studying psilocybin at this point, but the stuff that kicked all of this off was actually using psilocybin as a laboratory model for mystical experiences. And the striking thing about this, apart from the fact that people were having pretty profound experiences, was that there seemed to be long-lasting behavioral and mood changes, which led to resumption of this clinical work that was moribund since the 70s. And so I think there's a lot of challenging things working with the Schedule One drug, just in terms of like the bureaucracy, the logistics, government approvals, like how thick is your safe, et cetera. But there's also a societal there's a lot of people that are convinced that like this is really going to work, and there I think is some overly inflated expectations about how effective all these treatments are going to be. And so I think there's a scientific communication piece that's a little bit tricky to navigate when it comes to studying psychedelic drugs. They seem to be fairly promising, but we actually don't have all the information to know exactly how to use them and how to integrate them within existing um, uh, treatment.
Oh, thank you. Thank you for that. What about I guess I've flipped the table on you a little bit from Jenny talking about reductionist one compound at a time when it's the actual huge, plant material that's being ingested. In your case, you're doing a sort of a reductionist approach in humans with the one compound when many folks that are out in the common public that are utilizing psychedelics are doing it with mushrooms. And so do you have any comment just about the differences in what you think or might think the activities are or the complications are when looking at that?
So yeah, I mean there are other alkaloids in psilocybin mushrooms, and up to now it's not clear that any of them are meaningfully psychoactive. Um, and we use synthetic psilocybin. There are other places that are using mushroom-derived psilocybin. And I think there is, on the part of some sectors of the public, a great deal of interest in the entourage effect, this notion that like somehow the mixture of alkaloids that's found in the natural product is better. And to my mind, that's not all that likely. It's never been formally looked at, but none of the other alkaloids seem to be psychoactive as far as I'm aware. And so it does kind of I think a lot of people are actually surprised that we are using synthetic product and that it's not mushroom derived. And there's almost like an expectation that of course this would come from nature. So, but it's never actually been formally studied. Is the natural product in some way different or better.
Yeah, thank you. I think it's just an important discussion point, and really glad that you're here to share that. And I'm going to jump over to Dr. Babylonis and just get some input there because you are doing the more full spectrum, if you will, studies in humans and not looking at just isolated uh, things. So if you could share your pathway to getting to where you are and some of the challenges that you've faced.
Sure. Thank you. So, I became interested in cannabinoids similar to Dr. Wilkerson, was an opioid researcher by training. And so I did my postdoctoral fellowship with Dr. Sharon Walsh and Michelle Offwall, and looking at opioid use disorder, behavioral pharmacology of opioids, and then became quite interested, similar to Dr. Wilkerson, of the overlap between the biological action of those two. And just kind of started from there, and then have kind of continued on in both areas, and focusing now on the intersection of those two primarily. And I really love that question about looking at an isolated drug versus a whole plant or a full spectrum type of agent, because I think an interesting question is what you're trying to model. Are you trying to model the use that's going on in the public right now? And could that be a therapeutic option? Or are you trying to kind of nail down which agent could be the most therapeutic when given in isolation and at what doses? And so that's always a question that we grapple with, is particularly for our therapeutic clinical trials, is which do you decide? And so we have two clinical trials that are about to launch with our cannabis center. One is a double-blind, placebo-controlled, randomized trial for patients with cancer, looking at edible doses of cannabis for cancer-related quality of life, pain, insomnia, appetite, all the things, cancer burden collectively. And we chose to use what folks in the field would call like a more broad spectrum drug extract in those edible doses. And the rationale was to try to model what people are currently using, current consumption patterns look like. And you could kind of go either way, right? But that was our decision. And then we are also looking at a combination product of THC and CBD with those full array of minor cannabinoids as one of the dosing dose conditions for that study. And so I'm always really interested in hearing people's take on which option to choose. But in our clinical trials right now, we're going for a more broad net with cannabinoids.
Great, thank you. I think along that line, and I think we've talked a little bit about this, but knowledge gaps in all these different areas that we've talked about, either specifically or more broadly, when working with sort of plant-based medicines. And I don't know, Shauna, if you want to directly just sort of hit that first, since you're up, and talk about some of the gaps that are there in cannabis.
I think there's more gaps than there are answers in the state of the science right now. And so, as Dr. Wilkerson has mentioned, sometimes there's a really rich literature of animal research, but then trying to bring that over into a clinical population is very difficult. And I don't think that we have fully achieved that on any therapeutic outcome yet with cannabis, profoundly. to the level that it would rise to a formal clinical recommendation or FDA approval, obviously. And so I think there are more gaps than answers at this point.
Yeah, so that's really interesting. And then in trying to bridge some of that gap, Jenny, maybe you can speak to some of the gaps that are there from you already mentioned some of them, but just detailing particularly when you're working with psychoactive substances.
Sure, absolutely. So, when a big push in the basic science field, especially with regard to psychoactive substances that may have therapeutic potential, is basically trying to find a therapeutic window to where we can potentially see a therapeutic effect before we start to really see the psychoactive effects that might not be very, very useful in a broad patient background. So although, there's the personal use of these compounds for their psychedelic or psychoactive activity, in a broad patient spectrum, a broad patient background, you might not necessarily want a patient to experience those effects in a normal daily lifestyle, but you would potentially want those therapeutic effects. And so a lot of the things that we do on the basic science side is we do try to figure out kind of how broad or how narrow that therapeutic window might be. And so that's what we've really tried to look at. And so for some compounds, some natural products have wider therapeutic windows than others before where we start to see a really large psychoactive effect. And so another thing that we're trying to do to kind of get to that point to where, like Shanna mentioned, there's kind of that discrepancy between that preclinical literature and what's been found clinically is so as I mentioned before, we know that there are limitations with our preclinical models. So especially for pain and some models of drug addiction have very significant weaknesses. And so within our field, within the pain basic science fields and substance use disorders fields specifically, I can speak to the point that a lot of researchers are really trying to make the tests that we use more applicable to what's more kind of the things that would be going on in a patient. So obviously we can never perfectly model what a human goes through and what a human outcome would be, but basically trying to look at more translatable endpoints going from animals to humans is another big thing that we're trying to do to kind of minimize and kind of close those gaps.
Yeah, thank And it's interesting because in many of the cases, particularly in kratom or kratom use, there's a sort of mantra in the user side of the world, the human user side, that less is more, and that lower doses are actually more effective at producing uh, a therapeutic sort of endpoint that they're looking for, which is really hard to come back and bring back to a basic science laboratory and try to study from that standpoint. And of course, it's very difficult to understand if an animal is having a psychoactive event. So we have little things to measure, particularly in the cannabis area, like looking at uh, uh, the catalepsy models and some of the other assays that are there, but do they really capture what the human experience is? It's hard to say. And it sort of leads me back to Sandeep and talking about potential microdosing and how we hear about microdosing versus, a full-on sort of therapeutic or psychoactive dose of like psilocybin, for instance. Do you have any comments on some of the understanding gaps there or some of the limitations in looking at the differences?
Sure. I mean, there's two directions I could take this in. One is with microdosing in particular, there's the what does the science show so far versus what does public opinion look like. And so there's entire books written about how effective and amazing microdosing psilocybin is or LSD. And so far, the studies that have been done do not show that it's doing something that a placebo is not, with a couple of few exceptions, but it certainly doesn't line up with the perception of people who are microdosing enthusiasts. And I should add too that there's actually a fairly plausible risk of cardiac fibrosis from chronic use of any common psychedelic. So it's not there's still gaps in evidence though. It's never been studied in people with depression or a clinical condition. It's never been paired with something like therapy or meditation. So it's possible it still might be useful, but there's certainly nothing really that lines up with the impressive effects people report. But on the other end, the macro dosing, the big doses, and I guess I can talk about this from like a drug development perspective. The way that these trials end up shaking out is that people have fairly obvious and profound subjective effects that often end up becoming very personal and meaningful and lead to long-lasting changes in their thoughts or their meaning or insights about stuff that themselves seem to be therapeutic. And so there is something about this, it's a drug experience, even though it's wrapped in psychological, wrapping with meetings with therapists, etc. But it's a drug experience that appears to, at least in part, act therapeutically through something that looks like psychotherapy. And this is something that doesn't really neatly fit into modern drug development paradigms. And so there is a real difficulty in trying to figure out how do you study this, but how do you study it in a way that can lead to potential approval for a medical product?
Yeah, I think that's a sort of set policy and public health implications for some of these substances, and particularly the challenges of studying something that's Schedule One, and how, if you have any insights on, how do you move that needle, so to speak, with the regulatory bodies or policy making bodies, to show them evidence that there is therapeutic benefit, and that this should be reconsidered. And is there any type of rescheduling discussion going on for psilocybin like there is for cannabis?
There's not, just starting at the end, the rescheduling thing. I don't know. I've not heard of anyone seriously talking about that. And my understanding is that in order for something to be, if there's a drug that has some kind of effect that may be viewed as desirable, like it makes your mood better, that boom, that's considered as having an abuse potential. And if it is not an FDA approved drug, then it has no accepted medical value by definition. And so my understanding of what's going on with cannabis is that that definition is being kind of modified to include state level medical use. And so now you have accepted medical use, now it can be descheduled. And so there would need to be a reinterpretation of no accepted medical use to include something like, FDA has granted breakthrough therapy designation and is providing an unusual degree of support for the development of psychedelic medicines, and yet it's not actually approved, right? So I think if there were some modification of that, which I think could happen within at the executive level, then perhaps it could be descheduled. However, that doesn't really solve the issue. And so there are infinite psychedelic drugs. There's psychedelic drugs that haven't been made, right? Right now, our center is involved with a drug company who has made a new psychedelic. They've just sort of slightly modified the chemical, and boom, now it's not scheduled at all. DEA's never heard of it. And so it's still a pretty difficult study to run. We just don't have to get DEA approval. We just don't have to have um, put it in a enormous safe. But it doesn't. I wouldn't say it really solves all the difficulties of psychedelic medicine.
Sure. No, that's interesting. Thank you. Thank you for that. And I think the interesting aspect of this panel is we have sort of what you're doing, and then Dr. Babylonis with the cannabis work. Federally it's still Schedule 1 as well, and so maybe if you have comments on sort of how difficult that is, to work with when at the federal level it's one way and at state levels it's varying across the country, and those policies are looking to change, at the federal level. What kind of challenges specifically to that, but also just kind of any insights you want to share around that.
Sure. Lots of issues with Schedule 1 drugs, lots of problems. And so first, just to address like the discrepancy between state law and federal law, as federally funded researchers, we're held to federal law. And so no matter the state level policy, we're still held and beholden to Schedule 1 status for cannabis and related cannabinoids. And so one would hope that, legalizing cannabis in your home state would allow you to go to a dispensary and get high quality product to use in your trial. And unfortunately, that's not the case. I think some lawmakers have tried to make that happen, but it has not successfully succeeded yet. And so what happens then is you are forced to get drug from a federally sanctioned source for human research. And that requires both DEA and FDA IND approval. And I will tell you, all of our trials have been held up for plural years because of drug supply issues related to this. And primarily to more on the side of the FDA pathway. And so, yes, you could get, there are new suppliers that are cropping up that are DEA sanctioned, but they are not ready to give FDA approved type drug to a researcher. And so they might have drug, but it doesn't pass muster with the FDA because they don't have all the specs, they don't have all the research, not all the chem tox information yet. And so our group, and I know the group that Dr. Nyack works with at Hopkins, has just been the victim of this kind of regulatory structure. And again, I went with one drug supplier and they promised that they would have it hand delivered, and that company ultimately collapsed. And I got a new supplier, and that supplier was a year late supplying this drug. It took a 1 year longer to create the drug doses than anticipated. And so several of our studies have just been delayed by years. With just cannabis, right? This is not anything like fancy. It's just isolated cannabinoids. And the same is true with plant cannabinoids as well. And so it's a tough road, and there's a lot of setbacks. But once you have drug and you have your approvals, the world looks a little bit better.
Yes, I think it's a really great segue to go back to a totally unregulated substance, which is kratom. And Jenny, do you have anything you want to add to sort of working with a substance that's totally on DEA's radar? FDA has issues, working with a substance, sourcing it, addressing some of the same things, but from a basic science side, where we don't have the restrictions of regulatory barriers here, but what are some of the challenges and barriers with materials?
Sure, thank you so much for that. So, when we talk about animal work and preclinical work with some of these natural products, so although I've never personally had an issue obtaining any cannabinoids, even Schedule 1 cannabinoids, that's never been an issue so much for me. However, for the preclinical side, we don't have to get FDA or approval. We do sometimes have to have DEA Schedule 1 licenses, which I have, but instead what we work with is we work with a government contract facility called Research Triangle Institute, who supplies the research grade cannabinoids for basic scientists to work with, that are approved through the National Institute on Drug Addiction. And so I've had to work with RTI several times, and so even on the basic science side of things, minor cannabinoids, hard to isolate cannabinoids, kind of the thing we were talking about before. I've asked them, hey, can I get, so much, of a minor cannabinoid that's really, really pure? And I've gotten, well, we don't have that much in stock. We can give you this little trace amount. Can that be good enough for you? And so it is also a limitation on the basic science side as well. And so that's the part A to, I believe, what you were asking, Dr. McCarty. The part B is when we're working with an unregulated compound, unregulated plant like kratom, some of the issues that can occur. So when we first started working at University of Florida, we weren't quite sure how the DEA scheduling was potentially going to impact kratom because there had been a measure to schedule kratom as a Schedule 1 drug, and the different alkaloids in kratom as being a scheduled compound. And so we weren't quite sure how the regulatory landscape was going to look with regard to kratom. And so we, Dr. McCurdy and our group had very, very candid conversations of, okay, so how do we proceed if this drug does indeed become scheduled? Or now certain states are starting to delegalize kratom in a state by state measure. So what would happen if Florida, for example, were to make kratom illegal? What would that look like? What would we need to do in order to keep using kratom for our research purposes and the different kratom alkaloids for research purposes? So that landscape can change depending upon state law as well as federal law. So it's definitely something to kind of keep an eye on and keep in consideration.
Yeah, and I'll add a little bit that maybe Dr. Wilkerson didn't know about some of the struggles with particularly an unregulated substance that's up for potential scheduling. So I actually had kilos of Mitragynous speciosa leaf material when the DEA announced its intention to put it into Schedule 1. So I was going to be um, waking up in the morning and guilty of, having enough for distribution and trafficking, et cetera, et cetera. So I called and asked specifically, what do I need to do with this material that's potentially going to become Schedule 1? And they said, well, right now it's not under our jurisdiction, so we can't provide you any guidance. And I said, what if I wake up tomorrow morning and it's Schedule 1 and I've got 50 kilos of leaf? Am I going to go to jail? And they said, well, we don't even have a drug code to give you because it's not under our jurisdiction. We don't have any way for you to put this in place. And so finally, after many, many conversations around this topic with them, the best solution that we came up with was inventorying everything that we had and treating it like it was a controlled substance. And then making sure that all of our bulk material was being stored with someone that had a Schedule 1 license, so that in the case that it did become Schedule 1, it was in the hands of someone that was authorized to have other materials that were Schedule 1. And once the drug code became available, they would be able to capture that. And so that's a cautionary tale, I think, on any plant-based medicine that is emerging currently. I mean, I know there's other plant-based medicines that are being studied for some of these same potential uses in substance use disorder or mood disorders. And they're not regulated, they're not scheduled, they're not going down the pathway of a FDA botanical drug approval. So there's always this risk that studying a plant, although it may be benign today, suddenly tomorrow the DEA comes out with an alert or an intent to schedule because of whatever reason. So I think these are other questions or other things to really keep in mind, that when you start working with a plant that has psychoactivity or purported psychoactivity, that you really need to think these things through as if you are handling controlled substances, just for your own safety and your own protection. And so I don't know if anybody else has anything to comment in that space on the panel.
I would just have one quick comment insofar as even though kratom isn't scheduled at this point, it is still incredibly hard, if not impossible, to get a source for a clinical trial. And so I think that just kind of illustrates some of the issues that we've been talking about. Like, it isn't scheduled, but to get a pure source that the FDA will say, yes, you can go to clinical trial with this drug and get an active IND is just incredibly difficult at this point.
Yeah, and I can speak directly to that because we were involved in sourcing the materials for the FDA's SAD trial in humans, which is the report should be coming out hopefully at the end of this calendar year, but early peer-reviewed paper, hopefully early next year. And we're in the process right now of sourcing another batch for human abuse potential trial for FDA. And in fact, I was in a meeting prior to that, prior to this one, FDA scientists on that sourcing issue. And it's very complex, it's very complicated. And I think that it also goes back not only for the clinical aspect, even for the basic science aspects, because we have to verify that what plant material we are getting is actually what we believe it to be, number one. Number two, it has to be characterized and fingerprinted and make, authentic. We even go as far as doing DNA barcoding, and now we're doing full genome expression of the plant materials from authentic sources in Southeast Asia, so that we can actually fingerprint back where all these things are. But it's very complex. And then one thing that we haven't really talked about are some of the GMP regulations that these materials would have to meet by FDA standards for human trials. We want that same sort of level of protection for the animals that we're testing as well. So these materials have to undergo, bacterial testing, fungal testing, aflatoxin testing, mycotoxin testing, heavy metals testing. So there's a lot more to just sourcing a plant material and saying, hey, we're good to go. Even at the basic science animal level. So thank you for that.
Can I add one or two things to that really quickly?
Sure.
Okay, so I just wanted to definitely agree with that. So, as Dr. McCurdy mentioned, there are definitely some issues when you're testing preclinical plant derived compounds in animals. So, I'm not sure if Dr. McCurdy was trying to allude to this, but we had some significant issues with this when we were starting to work with the metrogynine and other kratom alkaloids in our animals. because basically we found that even though there's a small amount of other kratom related product in the originally isolated metrogynin, that small amount of other product actually ended up having a large physiological effect. And at first we weren't quite sure and so it took a little bit of time for Dr. McCurdy's group to really figure out, okay, what extraction technique is going to be the most appropriate for this material to where we get the purest, unadulterated from other plant alkaloids, the most purest compound that we can actually use to work with. And so, I mean, RTI for cannabinoids, they go through the same issues. So, for example, trying to source CBN from RTI, routinely their CBN, they even say, hey, there's a trace amount of THC in the CBN. We couldn't quite do a perfect extraction, so because there's a small amount of THC, you have to make sure this is kept under Schedule 1 conditions. So that is definitely a main, even on the preclinical side, a main concern.
Yeah, thank you for adding. Um, I am watching the Q&A box. There's nothing in there at the moment. So just a reminder, if you have questions, I know we're running out of time, but we want to be respectful. If there are questions, please go ahead and type them in. Otherwise, we'll continue carrying on and talking through things. And I think one of the other areas that I guess we haven't touched on is looking at any important sex or gender differences that we've seen in the use of plant-based uh, medicines. I'll just leave that open to the panel. Okay, hearing none. Um, any other topics uh, that you guys want to share that we haven't covered? Jenny, I'll start with you.
So, one issue that definitely pops up is we know that in the human world, that patients are not just using these natural products by themselves. So, as Dr. Babylonis and I have mentioned, we know that people use cannabis and opioids combined. We know that people are using, for example, kratom plus CBD combined. And so we know that people are using these drugs or these natural products in combination with other substances. And so a major area that's kind of become, I think, a cornerstone of the pharmacology worlds that we live in is to really try to figure out what these different drugs are doing in combination with one another, because really we cannot exclude natural products from a drug category. They really are indeed drugs. So what are these drugs doing in combination with other drugs that people are using?
Yeah, thank you. Dr. Nayak, you want to add anything to that?
Yeah, this is like not very well thought out, but there is sort of an odd feature of working on the medicalization side of drug or an intervention that's already being used by millions and millions and millions of people. Um, and that's actually very useful for us from an understanding perspective, but it's the political side of things that becomes fairly unclear. So there are many state level, kind of like what you saw with cannabis, but different. There's state level legalization, decrim initiatives, but also pseudo medical initiatives. And so I think I don't have any grand takeaway. I think it's just a very interesting environment to work in when you have so many different cross currents that are all ostensibly dealing with the same thing.
Yeah, so I think there's actually a couple of questions that have populated our Q&A box. So I'm gonna just go ahead and address one of these here. For those of you that are doing human work, how do you control for your participants that have used substances of interest in the past? Do you actually control for it, or just report that? Just any comment?
Yeah, I see that the rest of that brings up the MDMA stuff. So people who have taken, let's take the MDMA example, people who have taken MDMA and then are entering an MDMA trial can be assumed to be different in some way from people who've never taken MDMA or psilocybin or anything else. And so there's some ways of handling this. So some trials are, particularly early on, are saying we don't want anyone who's ever taken this. Others are saying we want to have it like the big phase 3 trials. I think they cap it at 10% or something like that. But ultimately, as with the case of the MDMA, you can also, the concern is that people who've taken the drug already are different in some way. Maybe they systematically do better, systematically do worse. And so you can also just look after the fact and see if that is the case. So with the MDMA results, people who'd gotten MDMA before didn't do any better or any worse than people who hadn't. So you have a range of ways of dealing with it.
Or Dr. Babalonis, you have any comments?
Sure, I'll just comment and say that I think it also depends on the question that you're asking, right? And so if you are looking at a human abuse potential study, you would want participants with some experience with the drug that you're testing or the drug class that you're testing, because if you use naive people, they are not going to respond to any of the abuse signals that someone who currently misuses that drug. And so I think it really depends on the question that you're asking. And so for the trials that we do that are human abuse liability trials, we do control for that use. And so, for example, we have a study going on right now where we're enrolling people with physical dependence on opioids. And so we have cutoffs for those that are pretty well established of how many days of use that they need to have, what types of use they are required to have. So if you are administering a drug in the lab through an inhalation route, you're always going to want someone who has been exposed themselves to that route so that you're not exposing them to the highest risk possible. But overall, yes, we do control for that and report it as well.
Yeah, thank you. And I guess I'll throw one of these questions at Jenny because it's specifically for a newer preclinical lab that's trying to get a Schedule 1 license. Can you talk about the process about applying for a Schedule 1 license and getting it awarded?
Sure. So it is, it can be a difficult process if you don't know kind of the hurdles to jump through. So if if you're a new investigator and you're wanting to use Schedule 1 drugs, it's kind of the chicken and the egg kind of scenario. Well, the easiest way to get approval to use Schedule 1 drugs is to have a grant that outlines the use of Schedule 1 drugs. Although usually you want to include the fact that you've had experience using Schedule 1 drugs to write the grant. So it's definitely the chicken and the egg approach. So what an alternative strategy can be to use non-scheduled drugs, non-scheduled synthetic drugs, to get some initial data collected for that initial grant submission. So basically, okay, hey, yes, I realize, in my main studies, I will be using Schedule 1 drugs here for proof of principle. Here are some other non-scheduled drugs that act at the same receptors that can produce similar results. So that's one strategy that is very, very useful for an early stage investigator that might not have that availability. The second route is if you have like an IACUC animal use protocol or an institutional safety protocol written up to where, okay, so maybe you've got startup funding and you want to proceed with using Schedule 1 drugs, go ahead and write up your animal protocols to use those Schedule 1 drugs, write up your institutional safety if you're doing cell culture work to go ahead and use the Schedule 1 drugs in that manner. So in order to actually get that DEA Schedule 1 license, you really have to show the agent that you have a commitment by your institution to allow you to do that research. So that's really the main key. So you have to have that IACUC protocol approval. You have to get that institutional safety approval in order to get the DEA Schedule 1 license approved. And so I had several of those things already lined up. So I actually was involved in a grant that had DEA Schedule 1 compounds being proposed to use. So I wrote up my animal protocols saying, hey, I'm going to be using these Schedule 1 drugs. And then I applied for my DEA license based upon all of those steps. So for me, it was a very clean shot to get my DEA Schedule 1 license. However, I went to go add on an additional drug to my DEA license Schedule 1. So you have to add each code, each DEA Schedule 1 code to your DEA Schedule 1 license as it is needed. So it's not like some of the lower schedule DEA schedules to where it's a blanket schedule. So if you have a schedule 2 through 5, basically any of those drugs you don't have to get additional approval from the DEA to use. You might have to have state approval, but you don't have to have individual DEA approval for each individual drug underneath that license. For Schedule 1, it is different. You have to have DEA approval for each specific code that drug code that you plan to use. So if, for example, I started out using just cannabinoids on my DEA Schedule 1, but then I wanted to also add in heroin to my DEA Schedule 1 drug list. And so I had to actually go ahead and get IACUC approval for the use of heroin in my studies before I could add heroin onto my DEA Schedule 1 list. And there was some confusion actually with my IACUC committee about, well, you don't have DEA approval for heroin on your list. And so I actually had to have my DEA agent. I had to send my DEA agent an email saying, hey, would you please contact my IACUC committee to confirm that this is indeed the correct process, that I have to have institutional approval for the use of this DEA compound before you will approve it on my DEA schedule. So he graciously, agreed to do that. And so he contacted my IACUC committee chair and had that conversation. And so it was approved. But it can definitely be a process.
Thank you, Jenny. We're overtime actually, so unfortunately, we won't be able to answer any more questions or address anything else. But I do appreciate everyone who's been able to attend, and I see we've had several drop off already. But I want to close by saying thank you to the panelists and thank you to everyone for joining us today. As a reminder, the symposium events are happening throughout today and Thursday, and the full agenda is available on the symposium website. The link can be found in the chat. Wish everyone a great day ahead and hope to see you at another session in the symposium. Thank you all.